This is an article by Ed Yong. Ed Yong writes for The Atlantic. He's a science writer. The title of the article is Landmark Study on the Origins of Alcoholism. For Marcus Helig, the years of dead ends were starting to grate. A seasoned psychiatrist, Helig joined the National Institutes of Health in 2004 with grand ambitions of finding new ways to treat addiction and alcoholism. It was the age of the neuroscience revolution, and all this new tech gave us many ways of manipulating animal brains, he recalls. By studying addictive behavior in laboratory rats and mice, he would pinpoint crucial genes, molecules, and brain regions that could be targeted to curtail the equivalent behaviors in people. But it wasn't to be. The insights from rodent studies repeatedly proved to be irrelevant. Many researchers and pharmaceutical companies became disillusioned. We cured alcoholism in every rat we ever tried, said Helig, who is now at the Linköping University in Sweden. And at the end of every paper, we wrote, "This will lead to an exciting treatment." But everything we took from those animal models to the clinic failed. We needed to go back to the drawing board. Helig doesn't buy that mice and rats have nothing to teach us about addiction. It's more that researchers have been studying them in the wrong way. Typically, they'll let the animals self-administer drugs by pressing a lever, which they almost always learn to do. That should have been a red flag. When humans regularly drink alcohol, only 15% or so become dependent on the stuff. Why them and not the other 85%? That's a crucial question. And you won't answer it with an experiment in which every rodent becomes addicted. Eric Augier, who recently joined Helig's team, tried a different approach, one pioneered in his former laboratory to study cocaine addiction. After training rats to self-administer alcohol, he offered them some sugary water too. This better mimics real life, in which drugs exist simultaneously with other pleasurable substances, right? Given a choice between booze and nectar, most rats chose the latter, but not all of them. Of the 32 rats that Augier first tested, 4 ignored the sugar and kept on shooting themselves up with alcohol. 4 rats is laughable, says Helig, referring to the study's small size, but 620 rats later, no one's laughing. Augier repeated the experiment with more rats of various breeds and always got the same results. Consistently, 15% of them chose alcohol over sugar, same number as the proportion of human drinkers who progressed to alcoholism. 15%. Those alcohol-preferring rats showed other hallmarks of human addiction too. They spent more effort to get a sip of alcohol than their sugar-preferring peers, and they kept on drinking even when their booze supply was spiked with an intensely bitter chemical or paired with an electric shock. That was striking to me as a clinician, says Helig. Embedded in the criteria for diagnosing alcoholism is that people continue to take drugs despite good knowledge of the fact that it will harm or kill them. Many lab studies treat animals as if they were identical, and any variation in their behavior is just unhelpful noise. But in Augier's work, the variation is the important bit. It's what points to the interesting underlying biology. This is a really good study, says Michael Taff, a neuroscientist at the Scripps Research Institute who studies drug addiction. Since only a minority of humans experience a transition to addiction, an approach such as this is most likely to identify the specific genetic variants that convey risk. That is exactly what the team did next. They compared the alcohol-preferring and sugar-preferring rats and looked for differences in the genes that were active in their brains. They focused on 6 regions, 6 that are thought to be involved in addiction, and found no differences in 5, but in the 6th we did, says Helig. And it made me smile, because I'd started out doing my PhD on the amygdala. The amygdala is an almond-shaped region that sits deep within the brain and is heavily involved in processing emotions. When Augier looked at the amygdala of alcoholic rats, he found signs of unusually low activity in several genes, all of which are linked to a chemical called GABA. GABA, G-A-B-A, is a molecular red light. Certain neurons make and release it to stop their neighbors from firing. Once that's done, the GABA-making neurons use an enzyme called GAT3 to pump the molecule back into themselves so that they can reuse it. But in the amygdala of alcohol-preferring rats, the gene that makes GAT3 is much less active and makes just half the usual levels of the pump. GABA, therefore, accumulates around the neighboring neurons, making them abnormally inactive. The consequences of this are unclear, but Helig thinks that all this extra GABA hampers the rat's ability to deal with fear and stress. They're naturally more anxious, which might explain their vulnerability to alcohol. He predicts it will take another 5 years of work to fully close this loop, but for now, his team has definitely shown that GAT3, the GABA recycling pump, is important. They took rats that prefer sugar and deliberately reduced the levels of GAT3 in their amygdala. This simple procedure was enough to convert those resilient rodents into addiction-prone, alcohol-preferring 15%ers. At this point, the team submitted their result to a journal, and the journal agreed to publish them, which was good news. But after Helig's long history with rat-shaped dead ends, he wanted to do one more experiment. Curing alcoholism in rats is not important, he says. What's important is what this looks like in humans themselves with alcohol addiction. As it happens, it looks much the same. Helig's colleagues examined post-mortem tissue samples from people who had donated their brains to research, some of whom had alcohol addiction. As in the rats, they found nothing unusual in 5 of the 6 brain regions, but in the amygdala, they found low levels of GAT3. Other scientists have found connections between alcoholism, the amygdala, and GABA-related genes. But by identifying rats that are particularly vulnerable to alcoholism, Helig's team has begun fleshing out the details behind these somewhat hazy links. It's a very significant study that will impact the alcohol research field deeply, says Jun Wang from Texas A&M University. Identifying GAT3 is not that important because alcoholism is controlled by multiple genes, but the team's new approach will help find those genes. It's a wonderful method for modeling human alcoholism. There are other signs that what Helig found is relevant to humans as well as rats. A decade ago, a French cardiologist named Olivier Amiessin claimed to have cured his own alcoholism by taking a drug called baclofen. That was met with skepticism, and there was no basic science to support his claims, says Helig, but there is now. Baclofen stops neurons from releasing GABA. If individuals with alcoholism aren't good at recycling this chemical, it might be possible to compensate by producing less of it in the first place. But baclofen is controversial. It's a terrible drug, Helig says. It's been tested in several trials to mixed results. 2 studies which analyzed the results from the trials concluded that the drug's ability to treat alcoholism is only slightly above placebo, and its growing use is premature, and it can be harmful. People quickly build up a tolerance to it, which prompts them to seek higher doses. They can experience severe side effects, and France has seen more than 100 cases of people inadvertently poisoning themselves with baclofen. Other drugs like benzodiazepines also exert their effects through GABA, but like baclofen, they're easily abused themselves. They're a good alternative for alcoholism in the short term, but they're not safe in the long term, says Lara Ray from UCLA. But Healig's study suggests that other chemicals which could influence GABA levels in more subtle ways might help people to control their addictions. Several such substances are in development, and Healig's team can see if they change the choices of their alcohol-preferring rats. It's just such an impressive breakthrough for the field of alcoholism, Laura Ray from UCLA adds, with real potential for therapies. At least they've isolated the amygdala, GABA, GAT3. If those levels can be controlled without harming the body or causing poisoning or using chemicals that create additional addictions, then they're onto something.